So thank you very much for that very kind introduction and the talk. We're running a little bit late, aren't we? So, yeah. Am I all right to talk for half an hour? You sure? Okay. So what I'll do is that in the spirit of adaptive trials, I'll adapt my talk a little bit and won't talk a little bit about meta-analyses because I've got it in brackets there. But I'll talk you a little bit about clinical trials and a more strategic approach to these to improve health faster. So my talk is going to be very non-technical, although I'm a statistician by training, very non-technical, and trying to rethink how we think a little bit about clinical trials and what we're trying to achieve with them. And what we currently do is ask with our clinical trials, does this new intervention improve outcomes for this disease? That's what we do. And we know, those of us who've been in the game a long time, one or two of you in the field tick that box, I think, know that this process takes decades, doesn't it? And by the end of it, we often find actually what we've done is spent a huge amount of money, depending on who you talk to. That's 1.5, 2 billion, 2.5 billion pounds sterling or dollars. And we often find we've failed, actually. We haven't made any progress at all. And I've written up there some diseases that we've failed miserably in for decades. We've made no progress at all in these diseases. Motor neuron disease, progressive MS, Parkinson's, dementia, pancreatic cancer, so on and so forth. You'll have your own favorite diseases up there that we've made little or no progress for absolutely decades, despite continually having a go at evaluating new products. And the basic fundamental problem in this process has been that actually, if you think about the phase 1, phase 2, phase 3 process, what we haven't found is that we've not had phase 2 signals. We've had lots and lots of phase 2 signals. That's not been the problem. Those phase 2 signals have not translated into improvements in phase 3 outcomes. And where we failed is at phase 3. Okay? And you know, failure at phase 3 of an intervention is a real problem, of course, because it's most phase 3 trials don't show superiority. I think those of us who have been involved in phase 3 trials in this audience will know that many of us have been involved in trials which haven't shown superiority of the new over the old. They cost a huge amount. They cost, if you talk to industry, they cost about 75% of all the costs of evaluation. And they're much harder to do. They cost many, many millions of pounds. Some, if you talk to industry, hundreds of millions of pounds. They take many hundreds, if not thousands of patients, and they take many sites and again take many years to do. So this is a big investment. And we fail. An example of this sort of failure of phase 3 after signals of phase 2 is given in Parkinson's disease on this next set of slides. A lot of you will be familiar with Parkinson's disease. A lot of you will know people with Parkinson's disease, a horrible disease. And this graphic shows you on the sort of the brown blobs showing you the phase 2 trials, giving you a signal. Then the time passes, and then you do the yellow blob, which is the phase 3 trial. And consistently, we've had many, many signals from the phase 2, all failing at phase 3. And this is repeated across all of those diseases that I showed you, and this is repeated across many, many diseases on which we've made little or no progress. And we know for Parkinson's disease, we've only got symptomatic treatments. So how can we think differently? Actually, it's quite easy to think quite differently in this arena. The first thing we do is we change the question. The question we posed is, does this specific intervention improve outcomes for this disease? If we change that to how do we improve outcomes as quickly as possible in this disease, you think in quite a different way. Think in a very different way. And that's the question, of course, that the public asks and patients ask. Bizarrely, not the question that we as researchers ask. So it's quite simple. When you pose the question like that, it's quite simple how you might do this in the framework that we have. You evaluate on many interventions at the same time. So you spread your bets. You focus where we're failing in the phase 3 bit, not in the phase 2 bit. Because we get lots of phase 2 signals. And we acknowledge that many interventions will not succeed. We know that. History tells us that. And we're not going to get that much better, that much more quickly. And we adapt the protocol as we go, from data within the trial that we're doing and from outside. Those are the basic principles. Very, very simple. And this sort of thinking leads to this idea, the idea of platform protocols, which has sort of gained a lot of news time nowadays, of course, and a lot more platform trials are being done. And under that umbrella comes the multi-arm, multi-stage platform trials, basket trials, umbrella trials, lots of other different types of designs. But the principles are all the same. Lots of interventions all at once, focus on the phase 3 stage, acknowledge that many interventions will not succeed, and adapt the protocol as we go. And bizarrely, though, a lot of the platform protocols that you see are done in the phase 2 setting. And that's not where the problem is. We get plenty of phase 2 signals. We just get more phase 2 signals a little bit more quickly with that sort of approach. And I don't know why there's this big emphasis on platform protocols in the phase 2. It's partly, I think, because it's something that people can do rather than what they should be doing. Perhaps we'll pick that up in discussion. So this is our traditional approach up here. We've got a control and a treatment A, and we do that trial. Usually, often find that treatment A doesn't work. We then do the next trial in the same disease. Treatment B doesn't work. Then we carry on. That's what we do, isn't it? A very simple idea is that actually, if you have control going all the time, and treatment A and treatment B and treatment C starting all together, at various points in time, you compare treatment A against control, treatment B against control, treatment C against control. Say, is it worth randomizing further patients? What's the evidence up till now? And then stop some arms, because if the evidence isn't sufficient to warrant further randomizations, you stop those arms and you add other arms as you go. Very simple idea. It's not hard to grasp. And this is the sort of the multi-arm, multi-stage platform trial idea. And as I say, many have developed for phase 2 trials. Huge number of methods have been developed for those. And as I've just made the point, these will have limited impact. They've had some impact, but not huge, because you're still going to fail a lot at phase 3. And we as academics, most of you are academics have actually a real opportunity in this space, because industry is not going to do these trials at the phase 3 level. At any given time, a commercial organization will only have one asset that they will be interested in evaluating in a given disease. It's of no interest in them to have multiple assets being evaluated for a given disease at the same time, and they don't do it. We in academia have a unique opportunity here and of doing these things in the phase 3 setting. And experience shows that. These sorts of trials are faster, they cost less, they facilitate recruitment. There's actually a bizarre side effect of this sort of design, which does encourage recruitment, and we can perhaps pick that up again in discussion of why that might be the case. And they're flexible, you can drop and add arms. And perhaps some of the first formal MAMS platform trials in the modern era are our trial in prostate cancer, which I'll talk a little bit about in this presentation. That's a randomized phase 3 trial, which started with 6 arms to start with. It was a 3-stage trial launched in 2005. First discussions about adding arms came in 2007, and more formally into a platform trial in 2010. So this is going back a long way. And since then, 5 new research arms have been added. And I'll show you a little bit about that. And the reason I show you that is really to show you what the power of this sort of approach and what we can do to change the game completely in these settings. At about the same time, folks in the US were doing the I Spy breast cancer work that was led by Don Berry and others. That was a phase 2 platform and launched in about 2009, about the same sort of time. So this thinking was evolved about then. Let me talk you through a little bit about the thinking we've done in prostate cancer. So we did this thinking in about 2000s and developed the first major formal platform trial and launched it in 2005. So that's nearly 20 years ago now. At that time, there are over about 1 million new prostate cancers a year. We hadn't changed anything in this disease for 40 years. Hormone therapy was the standard treatment for this disease, for metastatic disease with prostate cancer for 40 years. No change. Can you believe that? How did we get to that? How did we get to that stage? So we planned this. Median survival was about 3 years for men with metastatic disease. It's a horrible disease, and you're not going to live very long with it. So we started this platform trial in prostate cancer, and we evaluated a number of agents: a chemotherapy agent, a bisphosphonate, COX-2 inhibitor, radiotherapy, the new androgen receptor agents, and metformin, a repurposed drug, and estrogens. That includes old drugs, metformin, estrogens, repurposed drugs, and radiotherapy, so non-drugs as well. And this is the trial. I won't go into it in great detail, but you'll see this sort of plot quite often. This is the control arm A. These are the research arms, the 5 research arms we started with, B through to F, adding various arms over time. The blocks show you the accrual period. The dotted line shows you follow-up. The black blob at the end shows you the time point of analysis. So we started in 2005, October 2005, and we're now in 2024 up here, and we've analyzed a whole host of arms. And we've adopted, and I'll talk about the control arm in a bit, but you can see that's the design. 1 protocol, 10 research arms over a period of nearly 20 years. So this is the sum of it all. Oh. Why is that working? Why is that working? It should work. It's not working anyway. Let's take this. There should be a graphic that builds, but it's not building, so don't worry. So what's the sum of this all? Over that period of time, we've randomized 12,000 participants in this trial. Over that 20-year period, there's been 5 changes in standard of care. 4 is a consequence of Stampede results themselves. So the trial is giving you results in real time, and we're changing the standard of care in real time, both the control and the research arms. And the key thing coming out of this is we've got better survival. Median survival has now moved from 3 years to 7 years for the men with metastatic prostate cancer. And if you think of any other way we were going to do that over that period of time, I think you'll find it very, very difficult to achieve any other way of doing that than using this sort of design. I would like to have claimed that these were the first platform trials that were ever done. Of course, there's hardly anything new under the sun, is there? And if you look back, in 1952, the MRC did this TB trial, and it was a basket multi-arm, multi-stage platform trial. Can you believe that? In 1952. This was the design. And you don't need to think about, just look at the different subgroups of patients. It's pretty much the same graphic, isn't it? I'd like to say I'm pleased I discovered that, but there we go. So some of the key things that come out of these sort of designs is you have to plan for a 5 to 20 year time frame. You have to think long term. You've got to change the way we think about these things. The funding can come in stages and from different sources. And again, I'm happy to answer questions of how you manage all of that. Patients are absolutely key to their design, their development, and funding. Actually, without the patients, we would not have got this off the ground. And because everything in society, the way we're funded, collaboration, everything is stacked against you. So you need something to break that wall down. And there's the patients who do that. It's a big challenge for the coordinating center. There's a lot of work for the data monitoring to do. It's really challenging to develop, fund, and run. And there are things that you can do to help it run. I'll just talk about the last 2 points. Is actually you have to plan when you're doing this, plan for both success and failure. What happens if one of your arms is successful? What are you going to do with the control arm and the research arm? And what are you going to do if it fails in terms of, what are you going to say to the patients, what are you going to say to the doctors, and how are you going to release that information? So actually it causes you all sorts of communication and scientific issues as you go, which are good problems to have. These are really good problems to have, because you've got something successful on your hands potentially. And what we've found actually, if you build this sort of infrastructure, industry will come. We have industry, at least in this trial, and a number of our other trials, banging at the door trying to see whether they can get involved with this. But build it before you get them involved. And again, we can pick up why that might be the case in question. Are there any statisticians in the audience? Oh, one or two. Here we go. This is a field day for statisticians. I won't go into any of these points here. You can spend your whole career working on issues around these trials. So it's a lot of fun for statisticians. I won't go into any of those. Oh, there we go. Some more. So there's all sorts of design issues, and there's even more statistical issues that you can play with. And there's a stack of literature on all of this now. When you're doing these sorts of things, be very clear about the strategy and the goal. It's all about strategy and keeping the goal in mind. It's easy to get veered off into all sorts of different aspects of it, subsets of patients, subsets of data, biomarker, all sorts of things. You know how people get all very excited. The goal is to improve outcomes. How are we going to do that as quickly as possible? And what do we do to achieve that? Well, you do big things. You're doing a big project, you do big things. You drop research arms, you add research arms, you change the control and research arms as you go. From within, you can even change the primary outcome measure, you can change the sample size, and you can split the population into subgroups. If you plan to do that and you retain the integrity of the results, you can do all of that. A lot of that goes against standard clinical trial thinking, doesn't it? But actually you can do all of that. And this is just a slide showing you, on the left-hand side, for the statisticians, all the methodological papers that we've got out of this sort of design. And actually, this is a bit old now, so there are many, many more. And so, the CV looks good for that, from that perspective. And on the right-hand side, all of the trials, or not all, or many of the trials that have used this sort of design. Particularly, for example, the last lot at the bottom there, you don't need to read them. There are a lot of them in the COVID era. A lot of those have used this sort of design. And that's where this sort of design has really picked up a bit of pace and a bit of newsworthiness. And more than 80 randomized trials use this sort of MAMS platform design for some modest variation of it. As I say, disturbingly too many still in the phase 2 setting rather than the phase 3 setting. I'll show you how we've developed this thinking in a surgical setting, actually. So those who are surgeons, any surgeons here? No, probably no surgeons here. But we'll all know that wound infection after surgery, whatever type of surgery, is a major problem worldwide. It's a major issue for the patient because it's got significant morbidity associated with it, lots of hospital stay, and to some, a small number, potentially death. A major issue for health service, given the amount of surgery that's done around the world. And so the question here is, can we reduce the infection rates after different surgical progress? Now, we've made little progress in this area, and the guidance is is on a very poor evidence base. There's hardly any evidence base. There's conflicting recommendations, and there's just generic guidance of what people should do at the time of surgery to reduce wound infections. So we've designed this Rossini platform, which has just been funded by the NHR as a platform trial, one overarching protocol, and we've got this wonderful sort of frame for it. It's a basket factorial MAMS platform design. Yeah. But you'll see the ideas are very simple. There's multiple parallel, separately powered MAMS trials across different body types. So these are the body types: usually breast cancer, vascular groin, cardiac surgery, neurosurgery, amputations, and thoracic surgery. All different disease areas or different surgical areas, all evaluating of the order of 6 interventions in a factorial setting, so 3 by 2 interventions. Some are common. Some of those interventions are common across different surgical settings. Some are not. And, you know, the things that we could do there is how can we learn from one surgical setting that is actually recruiting more quickly than another if we see either an effective intervention or an ineffective intervention, which can we take it over to other surgical settings if it's effective? If it's ineffective and it's in other surgical interventions, can we learn from that and say, actually, it's not worth a candle in that other surgical setting as well? So you can do some quite clever things here. And these are the sorts of design considerations, the baseline wound infection rate written at the top, what we anticipate the absolute risk reductions are in the different areas, and the total sample size about 26,000 across all of those surgical sites. We're trying to bring this idea out more widely to lots of different areas, particularly in those areas where we've made little or no progress. So we're working with many others in cancer, infections, maternal health, psychosis, particularly in neurodegenerative diseases, MND, Parkinson's, progressive MS, and dementia, to try and change the game in those areas. Because if we want to make progress in those areas in our lifetimes, we've got to do something different. And of course, when you start thinking about this sort of idea, you say, well, people say to me, well, of course, you can only do such things when you've got lots of patients, when you've got a short-term outcome, and you've got large funding pots available. Of course, that's a requirement, isn't it? Well, I think it's exactly the opposite. Because if you've only got a few patients available, then actually you want to maximize what you learn from them. And this is a much more efficient way of doing things than you would if you had loads and loads of patients available. If you've got long-term outcomes, then if you're going to constantly fail with your current strategy, then it's going to take you forever to get anywhere. A much more efficient strategy is this one, because you'll find that you'll fail and in a more efficient way and more chance of finding an effective therapy. And finally, if you've got limited funding, if you want to use it most wisely, this is a much more efficient way of using that funding than if you were to do lots of separate 2-arm trials. So it's exactly the opposite. So I think when you should prioritize these sorts of trials or approaches is when progress has been slow, when a number of new interventions with different targets are available. The different targets that are important here, and again, we can pick up why that's the case in the discussion. When patients and enough doctors and researchers are on board, you don't need everybody. There's always wherever you go, whatever you do, you have always a group of naysayers. Hopefully those naysayers are not too large. And of course, when funders are on board. Funders have got to be on board. How are we doing for time? We're not bad. I'll just conclude, and I'll stop there after this one. I won't talk about meta-analysis. We need to make faster progress in a range of disease. If we're going to make progress in our lifetimes, we need to make faster progress somehow. This is one way. You've got to start planning for a 10 or 20-year horizon. You know that in some of the recalcitrant diseases that we have, that we're going to fail a lot, and let's try and do that in a strategic way rather than in the rather poor way that we're doing it at the moment. Researchers and funders need to change their strategies in a number of these diseases. I think thankfully some researchers, as I've shown you there, and some funders are changing their attitudes. Not all. And actually, the patient and public voice is key to this, because one patient partner asked me, why don't you do all your trials this way? And I couldn't answer that question. If you're able to give me an answer to that question in the discussion, I'd be very grateful. Perhaps we'll stop that one.